we get experimental. SciAnts. We're curious, non-judgmental.
I realize this is a mouthful of a title, y'all. It's a bit of a complicated title. However, quick breakdown of the title tells us that they did some genetic engineering of plants. Photosynthetic platform is just that they're using photosynthesis to produce something. And the thing that they're producing is sugars that are found in human milk. Why is this cool? I would hope, y'all, that just by looking at the title alone, you would be like, this is amazing. But catch with this. In the world, about 75% of all babies drink infant formula in the first 6 months of life. Whether it be as the sole form of nutrition, whether it be to augment with breast milk, they're drinking it. But it does not replicate everything. So if you look at the nutritional profile of what is in breast milk, not all of those elements are in formula yet. And that's because they are very complex sugars in order to be made, and they're just very difficult to synthesize on a mass scale that you can put them into formula. Most of these cannot be manufactured, or if they are, it's in such low levels and the yield is so small that they're unable to do it. They made a plant to do this. And in fact, here's what's crazy. It's tobacco. So Sam, it seems like from a genetic manipulation standpoint, it works really well and it produces things that are complex at a high yield, which is really, really remarkable to be able to do. So it's one of those things that it's the ideal thing to be able to use, which is truly remarkable. Proposing that once they get this plant down, you can get the extra sugars and nutrients and just add it to the formula. So when you're making that breast milk formula, like the synthetic, you're adding in the supplements that make it as close to breast milk as possible. So this would be an additional element that would be added in to the formula. My guess is it wouldn't be like the way they phrased it in the paper is that it's not like you purchase it separately and add it into the formula, but rather the formula when it's being made in the factory would have this element added in at the time of making it, so that you as the parent wouldn't need to do anything extra. You don't need to buy a separate supplement and add it to the formula. You just buy this new formula that has more things in there that are closer to breast milk. Like cereal has iron. Yeah, exactly, Sam. So it's just like making it even closer. I mean, I think it's a really, really cool system to be able to do something like that. So the way they did the genetic engineering is they actually made the genes that synthesize certain enzymes that make the sugars. So in order to make these sugars, it's not just that you have to have the plant make sugar, it's you have to have the plant make the enzyme that links the sugars together in a certain way. So that's what gives these sugars specificity. It's not the sugar molecule, it's the enzymes that link the sugar molecule together. And so the plant naturally doesn't have these enzymes, but by adding in the gene, it will then have the enzymes and be able to do the job that otherwise it was not able to do, which I think, again, super, super powerful of a technique. Now, they modified the plant to make 11 of these human milk oligosaccharides. So these human milk sugars, they modified to make 11. And you might be like, Balint, you just said there's 200 of these sugars, and it's only making 11? Like, pfft. Okay, no. Because there's 3 major groups of these sugars. So if you take those 200 and you try to sort them by feature, there's 3 major features. Of the 11 they synthesized, all 11 were represented with those 3 major features. That's why it's a proof of concept that they're able to do it first before they do more of that. So Sam, one difficulty is, of the 11 that they made, there has never been an instance before where they made all 3 groups in one organism. So the fact that they made all 3 groups of these sugars that are different structurally in one organism, that was the first time to be able to do that. Yep, and so Murph, the enzymatic gene that they put in was from a human. And so that's, isn't that wild, Murph, to think about? And this isn't the only example, right? But you can humanize a fruit fly. Like, you can take a human gene and put it in a fruit fly and it'll be functional. Because again, there's a lot of genetic similarity. There's 70% similarity between a fly and a human. Here, you're putting in a enzyme, and that's just a part of sugar synthesis, and that's a human gene, and you throw it into a plant and it's going to process the sugar. But it's just truly remarkable that it's processing the sugar in a way where like everything is just totally functional. And so it's, yeah, that human genetic element in there. I think it's truly remarkable. Again, like that something like this is able to function. The issue is when you make plants and a genetic line of plants, they're not always genetically stable, meaning that there's a bunch of recombination that might happen. It might act up in a weird way. It might lose those genetic elements that you put in. And so making stable genetic lines are not straightforward of plants. It's hard to make something with multiple components. So instead, what they ended up doing is making one plant that makes one human breast milk sugar, and it's called LNFP1, and it was optimized for it. And the reason they picked this one is because they have found it is very highly expressed in human breast milk. Like there's a lot of it in breast milk, and they haven't been able to synthesize it. Okay, so they were able to synthesize and optimize this one particular molecule, this LNFP1, which Fishman, you might be like, Balint, what the heck does this stand for? Let me tell you. All right, here it is. It is lacto-N-fucopenteose 1. In the name of it, it does have that pentose, so it's a 5 sugar compound that we can get from the name. The fuco is it undergoes a process called fucosylation. So Murph, you were asking about the enzyme. The human enzyme that was put into the optimized plant in the paper does fucosylation, which is a certain sugar modification that is on there. So that's one of the reasons why you had to pop that in. So that kind of fucosylation doesn't happen in the plant, but you put in the human gene and it'll end up processing the sugar in that way. So again, with this, why they picked this molecule, this LNFP1, is because it cannot be made at scale. They also did what is called mass spectrometry, which is they looked at the components of human breast milk and found that this component was really, really high. They found no one's able to synthesize this at any high level, and so they were like, let's use a plant to do it. And they made a ton of it. Right now, remember we said like 43 of those sugars are able to be made in labs. Those are being able to be made in labs by bacteria, so not by plants. But these more complicated things are like, maybe we can use plants to be able to make these single sugars. We make a ton of it, and actually the way that you get it is you just end up grinding up the plant and then added it into the formula, is essentially the way they do it. That's like the quick way of doing it. They're saying obviously for implementation and commercialization there'd be a lot to do, but that's their big goal there. I think it's a really phenomenal paper and like beautiful human ingenuity just to be able to do something like this. So Cricket, the popular media articles have been like, hey, this plant's making breast milk. Not entirely true. It's making the sugar components that are in breast milk that are difficult to synthesize in any other way.